to understand the anatomical background, which I'll leave you guys with the memorization of that. But I want to get into also the concepts behind how neurons work specifically. So let's take a look at our outline. What are we covering today? We're going to talk about the functions of the nervous system. We're going to take a look at the anatomy, just to give you an anatomical background of the neurons, as well as the supporting cast. So neurons are going to be the main cells that are working in the central nervous system. The supporting cells are going to assist in the other supportive functions that we'll talk about as well. And then we're going to talk about the specific thing that neurons do is to generate an impulse. And that impulse is a form of communication. And we'll be able to see how that impulse is generated based on the ions moving in and out of the cells. And before we even talk about ions moving in and out, we have to understand the whole concept behind the membrane potential, which is related to the charges of the ions that are present on the inside and outside of the cell. And then lastly today, we'll talk about graded potentials. These are changes in the membrane potential. Today we're talking about graded potentials, and then when we talk on Thursday, we'll talk about action potentials. So I'll help you kind of tie in a little bit about the difference between the two. Then when we get into Thursday, we'll talk about action potentials, and we'll talk about the main differences between these two, because it's critical for you guys to understand these. So as we look at our how the nervous system is organized, you're going to see that out of all the cells in the human body, when we look specifically at our differentiated cell, our neuron, you'll see that our nervous system is about 3% of our total body weight, which is not a very high amount of body weight when we look at it. But reality is our neurons, the entire central nervous system and the peripheral nervous system, use about 25% of our total energy. So if you think about all the calories that you burn, it's heavily involved in there. And why is that so? Well, it's pretty much guiding all of the primary mode of communication between all the cells. Our brain, our central nervous system, our brain and spinal cord are sending out information to every single muscle, every single gland, almost pretty much every targeted tissue that it intends to target. And then not only that, it's also receiving tons of information. Okay? And we'll talk a little bit more about that in our next couple slides. And if you look at the central nervous system is really just the brain and the spinal cord, where you can expect most of the neurons, those specialized neurons, specialized cells that we were talking about, are located in the brain itself, as well as the spinal cord. You'll be able to see the same types of neurons. Now, in the peripheral nervous system, this is everything outside of the central nervous system, including all the nerves, things that come and project away from the central nervous system. We'll be able to review some of the spinal and cranial nerves, and know as a precursor you do not have to memorize those. We're really just talking about the way the nerves work. We'll talk about ganglia, the clusters of neuron cell bodies, and then plexuses. These are just a whole cluster of neural networks that work together, sort of the, if you ever looked in cellular, not cellular biology, but biology, when you look at some of the early insects, you can see that they don't have brains, they have plexuses. These are just a bunch of neural networks combined together. Now, I pointed out earlier that with the central nervous system, we saw information come in, and then we see information coming out. So let's take a look at an example of that. And in my example here, what you're going to be able to see is a car coming towards somebody. And so what I have for you on the next slide over is stuff that we're going to do together. This is a brainstorm activity. I'll have you guys pair up or group together, or even just give yourselves a couple minutes to kind of think through this exercise. And what I'd like you guys to think about as we go through this exercise is to think about, I totally forgot my pen. I need that today too. Will erase. So let's see what I can do. That's fine. Okay. So in this example, I want you guys to organize things in your head about things that you would, see how this is going to work, things that you would see. Okay. This isn't going to work out very well. Things that different reactions that this person's going to have. It's not going to work out. And we'll think about things that this person thinks about. Okay, great. So here we're going to see this image, and we'll play it for you. I'll play this quick video. Here you have a guy walking around. He sees a car almost hit him, and then he completely avoids it. Just for fun, let's play that back again. He's walking, completely avoids the car. The best part, he just literally checks his shoes and walks out, all right?
Where's that guy running? He doesn't even ask the guy if he's okay.
I have no clue.
He's like, oh God, I gotta go.
Okay, so think about things that this person is going to experience. What things is he going to sense? Not only the things that he's going to see. Okay, so think about things that he's going to see. Well, we can all sort of do this together. Then think about the things that he's going to think about as this is occurring. And then his reactions afterwards.
Professor?
Yes?
Would this work?
Let me see, does it have a pen? Let me see, thank you. Oh, it works better. Thank you. All right. So what are the things that our friend there sees or experiences? What sort of information is going into his head?
Cars coming at him.
So he's seeing the car coming, right? Car. Drive with the biggest crayon here. Car approaching. Okay. What else?
Sees the driver.
Sees the other driver. Okay. Another driver. Do you think he's able to hear things?
Tire screeching.
Tires screeching. Horn. Okay. Now, what do you think is happening in his head at the point of time, or even right before the incident, to the point that incident just barely misses him?
Move out the way.
Move out the way, right? Move.
I'm going to die.
Going to die. Low vitality at that point. How about just taking in that information? How about the thought process of, oh my God, that's a car about coming in my direction, about to hit me. And what I'm trying to get at is he's taking in information, right? The information he's taking in is sensory information. He's taking a look at the car approaching his way. He hears an accident, presumably down the way. That car looked like it hit something. And he also probably hears the tire screeching. He realizes, there's a car, I need to move. Okay. And then what are his reactions?
Kind of jump and
Jumps, right? Jumps out of the way.
Mm-hmm.
So some motor skills in place. What else is he doing?
check his shoes.
Checks his shoes, okay, all right, checks his shoes. What else? How about things that are not in his control? What part of your body do you not have control of? What would be a response? What happens if... racing heart. Thank you. So increase in heart rate. What else?
Increase in respiration.
Increase in respiration rate. Okay. Probably sweating a little bit.
Sweating.
Do you think his stomach is sitting there digesting his food at the same time?
No.
Maybe not.
Decrease.
Maybe we're going to see something turn off. Things get turned on and things get turned off. I don't know. I'll just throw in something. Maybe he screams. I don't know if he does. Doesn't really look like he does.
His adrenaline increases.
Adrenaline increases, so we'll start to see increases in adrenaline. With all this right here, out of all the days, I've never forgotten that pen, and today is the day because I do the most writing in this class. So out of these things right here, we'll see the jumps and screams. we can divide those reactions into two types of things. Voluntary and involuntary. Right? Things he has motor control of, and things that he does not have control of. Things that are involuntary or autonomically controlled. And you'll be able to see that in the next couple slides. So another way of looking at our left column here, see, thinks, and reaction. The way we can divide this up is that the top line represents sensory information. And this is information sensed by the senses that this person has, whether he hears, whether he smells, sees, touches, tastes. The next section is going to be the integrative sensor. And just in case I can't write that out, but you'll see the functions on the next slide over. Integrative, which has the term integrate, and what do you think the term integrate means? Kind of something together, right? If you don't know what it is, what do? You just kind of shrug your shoulders. You look it up. So if anybody wants to go in and actually look up the definition of integrates, and just go on Google and search out the term integrate, let me know. And that was the integrative center. And then while we're looking up that information, we'll look at that last one. The last one represents motor reactions. Did anybody find a definition yet for integrates? What's that?
To combine things?
To combine things. So the integrative center is combining not just things, but information. And what type of information is being sent to that integrative center? Sensory information. So what we're doing is we're taking this information in the sensory side of things, which we'll talk about in our next couple slides, but you probably already know his eyes, his ears, his nose. All the sensory receptors that he has is conveying information. Are the eyes doing any of the sort of thinking that's going on? No. All it's doing is just taking in information. It sees light patterns change, light increasing, and it's going to send that information over to the integrative center, where the integrative center is taking in sensory information from different pathways, integrating it together so that the integrative center can come up with, and here you can see the integrative center, processing that information. Processing of information from all sensory organs coming in. And what it's going to do with that processing of information, it's going to make decisions about it. Those decisions earlier that we saw, to jump, to scream, even things that he doesn't even have voluntary control of, increase of heart rate, increase of respiratory rate. So the integrative center has the processing of information, and you'll see the term perception here. And this should be clearly differentiated between sensory, which we know is the information that's being carried by the senses and transmitted over to the integrative center, which is doing perception. Perception is your sort of conscious awareness, the integration of that type of sensory information that's coming in. To give you an example of something, you're constantly sensing information, maybe the temperature of it. But the integrative center is realizing the temperature in combination with maybe other sensory things. What's happening with your skin? What's happening with your overall core temperature? What's happening to your blood pressure? All those things are coming in together. And this is the perception of information. If the perception of information, that's light entering my eyes, the integrative center is saying that's too bright. So what happens in the integrative center is processing, perception, making decisions, and something that's also not mentioned here is memory. Memory is extremely important because we got to store that information so that we can learn next time. And should be fairly obvious that the central nervous system is doing all that memory, storing that information so that it can improve the connections later on, which we'll learn about. So we see that the integrative center, is pretty much all the central nervous system, where we're going to see a lot of inner neurons. And I'll talk about this term in a little bit, inner neurons. And you see that 90% of the neurons, going back to our cells right here, looking at the organ structures, the brain and the central and the spinal cord are all consisting of our inner neurons. And hold on to that thought because we're going to go into that in a later slide. And then motor being the last of that reactions. You'll see the reactions that take place. And these are the decisions that were made by the integrated center becomes formed as a motor command of some sort. And that motor command, motor meaning motion, could be the contraction of a muscle. The muscles involved to get our guy to jump out of the way. The glandular reactions, the secretion of hormones as endocrine functions, which we'll talk about later. The secretion of sweat as an exocrine function. Probably some tears as well. Okay? And when we look at these are motor or efferent neurons. So here you see afferent information going into the sensory. We see efferent moving out of the integrative center. This is the affect and the effect. Again, one of the quiz questions from last Thursday. And those targets, muscles, glands, and quite possibly other neurons. And so this is kind of the example that we talked about in the first day of class, where we had sensory information. During that whole feedback loop, you're going to get sensory information going into the example that we talked about with our heating unit or air conditioner. The thermostat would relay sensory information to the control center, which we know is the integrative center, which is the central nervous system, where it's going to do the processing of information. It's going to make a decision. Is the temperature too high or too low below this set, or above or below this set point? And then it's going to send that information out the efferent pathway over to the heating unit to turn it on if we're below that set point. So that efferent pathway or the motor pathway would be everything out. Yes?
So is the sensory and integrative in the CNS and then the motor in the other one? Or are they both like in?
So where that would take place, and we'll go into more of that once we get into the nerves, that sensory information will take place outside, away from the central nervous system. And it's conveying information to the spine, over to the brain, where our integrative center is. And then the efferent pathways are going to be the nerves that are sending information out, whether it's targeting your fingers, your toes, any other muscle, or not just skeletal muscle, but any of our smooth muscles as well. Okay. Did I answer your question? So let's take a look at the divisions of the nervous system. Here we can see how it's divided up, where we're going to have our receptors, all part of the sensory pathway, the afferent pathway that leads into the central nervous system, which is our integrative integrated center. So this is sensory, this is integrated. And then our motor pathways are going to be out this way right here. This is the efferent pathway, as opposed to afferent, which conveys information to the central nervous system. And then we can divide the peripheral nervous system, which is the pathway outside of the central nervous system. We can see how it's divided up between the somatic nervous system, which is going to govern the contractions of skeletal muscle, and the autonomic nervous system, which is going to regulate all the things that person didn't have control over. All the autonomic functions that raise the heart rate, raise the respiratory rate. And here, the targets here, the effectors, which is the intended target of the central nervous system, can be smooth muscle, cardiac muscle, or any other glandular stuff. So that's our way of looking at it, the afferent versus efferent division. Now let's take a look at it from a pretty much the same perspective. But here what we're doing is we're dividing it up a little bit, in which we're able to divide information that goes into the integrative center and information that goes out of the integrative center. So here you can see our 4 specialized senses, which we'll talk about next week. Eyes, ears, nose, and tongue. And then we're going to take a look at other sensory organs that are located out in the peripheral nervous system that will also feed in somatic sensory neurons. These are neurons that govern the position of the body, the joint angle, things like that. Conditions of the body, or the musculoskeletal system. And then visceral sensory neurons are sending in information from the internal organs themselves. What is the blood pressure? How much cerebrospinal fluid is there? What's the pH of the blood at the time? How many what's the extracellular, what's the concentration of potassium in the blood? That information is being sent also into the integrative center, where this represents our special senses, and this represents our general senses, specialized senses, because they are coming from specialized sensory neurons. Now, we're not going to be talking about this until next week when we get into our sensory lecture. We'll talk about our general senses as well as our specialized senses. Again, we'll reserve that for next week. What we're really going to be talking about is the integrative center and the efferent pathway, information that's traveling out of the central nervous system. It's a little bit backwards, but it's perfectly okay because we'll wrap it up all together for you. And just like I mentioned earlier, somatic motor neurons are going to be targeting the skeletal muscles. These are voluntary things, as opposed to autonomic, which is going to govern the involuntary things. And here you're going to see our division, our sympathetic and our parasympathetic, 2 of which are going to work against each other. They're going to have typically exact things. So one thing will raise your heart rate, the other thing will lower your heart rate. And so they work hand in hand to keep us, again, within that window of balance or homeostasis. So with that, what we're going to do is we're going to take a look at the cells. The major key players are cells in the central nervous system, of which we're going to look both at the central nervous system as well as the peripheral nervous system. And as we learned in last Thursday's lecture, one of our specialized cells were the neurons. So here we take our basic cell, and that basic cell is going to express the DNA that's going to provide these other structures that are going to give its function based on the characteristics of the cell itself. And so before we talk about neurons, let's talk about the neuroglia. And neuroglia, we're going to be able to see are the supporting cast. And you're going to see the different ones that we have here. And I will have slides on these. I'll just touch on the general gist of what they do. I'll leave you guys to do the rest of the memorization. But nonetheless, let's take a look at how that's organized in the tissue layer. So here you can see the cells themselves. We'll be able to take a look at how those cells operate. And then we're going to combine them together under the tissue layer. And in that tissue layer, you're going to see the outer layer, the pia mater. This is the one of the layers of the outside of the brain. And if we were to take a cross section and slice all the way through down to the cerebrospinal fluid, you can see that there's another layer over here. And this is going to be our ependymal cells, which we'll talk about in just a little bit. And everything in between are all the types of cells that are found in that central nervous system. And we'll talk about these. Here you can see the capillary running through that's providing nutrients to those cells. And here's our example of our neuron, another neuron, and then you'll see all different types of cells. So what exactly is a neuroglia? Neuroglia is about half the volume of all the cells in the central nervous system. That means if you were to take a look, you would see that it's pretty much half and half, neurons versus neuroglia. Not necessarily in the number of cells though, I'm just talking about volume. Why? Because neuroglia are much smaller than neurons. In fact, we sometimes have about 5 to 50 times more neuroglia than we have neurons. So neurons, you would have known, are the ones that we pretty much have a set amount of. If you ever hear like going to college, you're going to pop some neurons. Yeah, sure. Those neurons, after damage, if they can't be restructured, they're gone. But we do have many of the neuroglia. A lot of text really just looks at the neurons, but the neuroglia are heavily involved in the supporting the central nervous system. So what do? Well, these neurons, these neuroglia, can undergo cell replication, meaning they can continually grow. And each of these cells have one specific function in order to support our neurons. We're going to be able to look at the types that are found in the central nervous system as well as the peripheral nervous system. The first of which we're going to take a look at, if we can see in the next slide over, here you can see the neuron itself in yellow, and the first thing we're looking at are astrocytes. And if you think of the term astrocytes, you'll see the prefix astro, which is star. Okay. And in this case, these astrocytes are the largest of them. And they're the largest because they have projections from the cell body that wrap around the capillary. And so it serves to protect the blood that flows through the capillaries from the surrounding extracellular fluid. And it does this in order to keep those separate. And this you may have heard before, it's called the blood-brain barrier. And the astrocytes govern that. They help separate stuff that's in the blood that should remain separate from the cerebrospinal fluid. So as I mentioned previously, certain drugs cannot pass through the cell membrane that is wrapped around the capillaries. Not only that, you'll see that the astrocytes also provide structural support, meaning they have these antennae that reach out and grasp onto the neurons and help anchor those in place. Other things you can see on there, what does it do? It does pretty much all of these right here. Make sure that it has an appropriate amount of gases, amount of O2, oxygen, as well as CO2. It makes sure that it has the right amount of nutrients, glucose, supplying, making sure that it maintains the concentrations of potassium as well as sodium and other ions that are involved. And then not only that, you'll see the last of it, astrocytes help kind of clean up the excess neurotransmitters that are floating around. So it does help clean up a little bit. Now oligodendrocytes, on the other hand, will wrap around the neurons. And they wrap around specifically around the axon, which is one of the major projections of that neuron. So while in the first slide we talked about astrocytes that wrap around the capillaries, it's the oligodendrocytes that wrap around the neurons, and we'll learn a little bit why. Oligodendrocytes will send out these projections and cover the axons in myelin sheathing. And what exactly is myelin sheathing? Well, it's this fatty tissue that wraps around, and it serves to insulate the axon. So think about your cables, for instance, the cables that run in your house. They're wrapped in insulation to make sure that the energy that's transmitted through is efficient. And it also makes sure that it doesn't cross-communicate with any of the other cells. So this helps provide that insulation. And what you'll learn later on, which we'll mention again later, is that this is actually going to help speed the communication as it flows through.
So the myelin sheath is composed of oligodendrites, right?
The oligodendrocytes are the cells that produce the myelin sheath. So they'll send out these arms and they just wrap that myelin sheath around.
So the myelin sheath is composed of it?
Yeah, exactly. Now the microglia basically are the cleanup crew. How do we know that? Well, you'll see that it is responsible, and it uses that term that I used before, phagocytosis, which is the eating up of cells. Any dead cells, any dead neuroglia that it needs to destroy, it'll come in and take it, it'll phagocytose it, meaning it's going to come up and swallow it. It's going to break down all those components, bring them back down to their macromolecules, our carbohydrates, our proteins, amino acids, phospholipids, and help regenerate new cells from such. The next one, the next layer, ependymal cells, this was what I talked about at the very bottom. And this is the area that you're going to see. So if the outer layer was the pia mater, once we get to the inner layer, this is going to be the one that's lining the ventricles. If you remember in anatomy, the ventricles are what's going to contain the cerebrospinal fluid that flows through the brain structures. And these cells, these ependymal cells, are a single layer. So going back to our tissue layer, do you remember there was two different types? There was simple and there was stratified. Since it's a single layer, this represents what? Simple. So it is one single layer of epithelial cells, And if you look at the shapes of them, they can be either cuboidal or columnar, depending on which epidermal cells we're looking at in the location of the brain. But for the most part, that single layer, all they really do is produce cerebrospinal fluid. So these are specialized cells that will generate that fluid that is secreted out into the lumen. Again, that term lumen, outside of the cell. So these cells support, let me ask you this, is it an exocrine or endocrine function? It would be considered an exocrine. Why? Exocrine is dumping outside of, so it doesn't necessarily mean it has to be outside of the body like a sweat gland or tears, something that dumps into a lumen. The endocrine are the epithelial cells that you would see that are wrapped around in capillaries. Again, we'll talk about the endocrine type of organs when we get to our endocrine lecture. So it's dumping CSF out into that cavity. And on top, the apical side, the top of that epithelial cells, are going to contain cilia that are going to help contract and move and sway like this in order to move that cerebrospinal fluid in the direction, help circulate that throughout the ventricles. So what exactly is a cerebrospinal fluid? You'll see that it's almost consistent with that of plasma. It doesn't have many cells in it, but basically a liquid that not only protects, it cushions the brain around, but it also carries all the nutrients, carries all the gases that are necessary for those neurons to operate, as well as the glia that help support those neurons. Again, more that we'll talk about in our central nervous system lecture. Now, in the peripheral nervous system, this is where I'm kind of zip through real quick. Satellite cells are the ones that surround themselves around the neurons. And Schwann cells are the ones that wrap around those axons in order to create that myelin sheathing. Just like the oligodendrocytes that you learned in the central nervous system, the Schwann cells are present in the peripheral nervous system, again, outside of the central nervous system. And all of these contribute, again, are support cells that contribute to some function, whether it's cleanup, support of the cells, and then regeneration. So if there's any breakdown of the neuron structures, here you can see Schwann cells kind of help build that along. So one thing I can just kind of talk about that we're talking about the myelin sheathing. Yeah, no, I'll get to that when I get to that section. Let's take a look at our neurons. So looking at our neurons, the main characteristic of neurons is that they are going to exhibit electrical excitability. And again, we'll talk about how that's generated when we look at the membrane potentials. And again, that's related to the concentration of ions on the inside and outside. There's a difference in the ion concentration on the outside of the cell as well as the inside of the cell. And so these neurons can fire. And we'll talk about that firing of an action potential at the end of today's lecture, and which will be leading into next week's lecture. Other than that, they respond to information that's being sent to that neuron. So you'll see different sections of it. You'll see the dendrites, you'll see the cell body, the axon, as well as the synaptic terminals, which we'll talk about in the next slides over. When we actually look at the cell body, here you can see the bulk of the cell. This is the main area that's going to contain the nucleus. And it's going to have all the same organelles that we talked about earlier. Inside the nucleus, you'll find the DNA. You'll find Nissl bodies, which are combinations of the endoplasmic reticulum and free ribosomes, that their entire purpose is to create those proteins that we talked about last week. And so these cell bodies are constantly producing things. Not only are they making more organelles for the ones that are failing, but they're also making proteins at the very end, at the synaptic bulbs, which we'll talk about. And those proteins that are going to be assembled are going to be our neurotransmitters that are involved in the synaptic communication. Again, something we'll talk about on Thursday. Now, the neurofilaments, we'll talk about some of the intermediate filaments that span the entire length of that neuron. And those are going to contribute not only to the shape, but you'll also see other functions of those filaments that serve in moving stuff around that neuron. Now going back, we'll see the dendrites. Here the dendrites are going to be these antennae that stick out of, they can either stick out of the cell body, or we can have specialized structures, which we'll see later on. But dendrites serve one purpose, and they receive information. These are called dendrites because the original dendrites, there's a Latin term for it, meaning little trees. And they're called little trees because you can see the spanning of these dendrites that extend out of the cell body. And they receive information. So they receive information that will get sent over to the cell body. And not only do you see a lot of them, but you also see a lot of branching. And that branching allows to increase the surface area at which our neurons can absorb information from adjacent neurons. And then if you were to take a look at the cytoplasm within the dendrites, again, you'll see things like missile bodies for the production of proteins. The proteins in this case would be the receptors that are built on the cell membranes. Lots of receptors that are being built. Now the axon is the part that's going to be conducting an impulse away from the cell body. So the information, the direction of travel of information is going to be from the dendrites to the cell body down the axon. And the axon can be the longest part of the neuron. And if you were to take a look at the axon, you can see that it's going to not have any of the missile bodies that are present in the cell body. So none of the protein manufacturing that's going on. A couple things I want to point about the axon. Here you'll see the myelin sheathing that's going to be present, that's served by the oligodendrocytes in the central nervous system, and the Schwann cell in the peripheral nervous system. So they come in and they wrap around certain sections of the axons, of which in between the exposed areas that are going to contain the neuron axon itself are going to be called the nodes of Ranvier, which we'll talk about. The things that are significant about the axon, you'll see the area called the axon hillock. The axon hillock. And this is the most sensitive area of the axon. The axon itself is going to now propagate that signal down the length of the axon, and it's going to make its way over to the terminals. And here you'll see axon terminals, or synaptic bulbs, or synaptic terminals. This is the very end of the axons. And within those, you're going to see these bulbs, these swollen bulbs at the very end that's going to contain neurotransmitters that are released by the axons. Other things that you would also see would be axon collaterals. These are side branches off the main axon. So axons don't have to go from one. They can actually branch out into another axon that can get sent to another area. So there's some branching that happens in the axon, but where you would see most of the branching would take place in the dendrites. And then the last thing that those terminals terminate on, they can terminate on muscles, glands, or again, other neurons. So looking at stuff that moves, as I was talking about earlier, things that are produced in the cell body, the proteins that are required to create those neurotransmitters are eventually going to have to make their way down to the synaptic bulbs. So let's see this highway of information. How does that travel through? You're going to see two types of flow that flow down the axon that are able to transport those proteins from the cell body down the axon. And you're going to see slow flow and fast flow. Slow flow is really just the movement of cytoplasm, or here you would see the term axoplasm. That's basically the axon's cytoplasm. And the direction of that movement is a very slow, unidirectional movement down to the axon terminal. So this is just the movement of stuff as it makes its way. It takes about 1 to about 5 millimeters per day. Not only is it moving the proteins, but it's also moving mostly the stuff that's dissolved in the cytoplasm. It's moving amino acids, it's moving gases, moving glucose all the way through. Now the other section on here is fast axonal flow, where this is moving through the use of microtubules. Again, the cytoskeleton. The microtubules serve as a highway for the organelles to be moved down. It's the whole structure that moves them along and moves the stuff that's also attached to the organelles. So this allows back and forth movement from the synaptic bulb, or the synaptic bulbs, back to and from the cell body. So here you would expect to see bidirectional fast movement to exchange with that. Now, there's certain diseases that are characterized by damage of that axonal transport. And one of the examples that you can see here is tetanospasmin, which is a toxin produced by the Clostridium tetani bacteria. In other words, a condition known as tetanus. So in this case, this tetanospasmin that's produced by this bacteria comes in and targets our neurons. And what they do is that, anybody familiar with tetanus? Well, you know you need to get a tetanus shot every 10 years, right? No. Well, this is a bacteria that's actually found in soil, it's found in dust, it's found in manure, and typically you would see like dirty nails that have rust on them. If you ever get stabbed by one of those, they would typically say, "Have you ever had a tetanus shot?" Okay, if not, they'll just give you an extra tetanus shot. It's the one that hurts. It hurts quite a bit afterwards, not during the shot, but afterwards. But nonetheless, that vaccine that they're giving you is tetanus, and what that actual spasmin is supposed to do is it's supposed to disrupt those motor neurons, the neurons that are involved in motion. And what it does is it causes a constant contraction of that muscle, a term that you probably would have heard called lockjaw, causing the muscles of the jaw to be extremely stiff and hard to move. And so what this toxin does is it actually interferes with the release of neurotransmitters that inhibit muscle contraction. So this, in order for me to describe, I have to draw what the neurons look like. So this targets the muscle. So here, and this is typically how I'm going to be drawing my axon. Here's our cell body, this is the axon, and this is the axon terminal. So we know the direction flows to the right. So this neuron, which you'll learn later on, is called a motor neuron because it targets specifically the motor function of the muscle. And so this is always going to cause contraction of the muscle. But in order to turn this off, there's no off switch. We're going to be looking at inhibitory neurons. And they come in to turn off our motor neuron. So here I'm drawing a neuron. This is an inhibitory, INH, an inhibitory interneuron that comes in and turns off that motor neuron to prevent it from squeezing. So to think of a contraction, any sort of muscle contraction, you can cause a contraction, and then for you to turn it off, to actually actively turn it off, is an inhibitory interneuron that comes in and turns off that neuron to stop it. And so if that bacteria or that toxin comes in and damages this, we have no way of regulating that excitatory motor neuron. It won't be able to turn it off, and therefore that muscle is in a constant state of contraction. So typically where you would see these, you would see them affecting the voluntary muscle. So it's not uncommon for people that have tetanus to start affecting the neurons that target the muscles. And there's obviously a cause of concern for that toxin to affect the central nervous system. Because if it starts affecting the central nervous system, those inner neurons, it can start to spread to the other neurons, which obviously is not a good thing.
So then if we have tetanus, we won't be able to move, basically with contractor, right?
Yeah, so some of the areas that are most sensitive would be around the neck and the jaw. That starts to affect it. And eventually, if the bacteria starts to spread, it starts affecting more and more of your motor neurons. It can start affecting the voluntary motor neurons, the ones around your organs, and seize them from functioning.
similar to like meningitis?
Meningitis would be a viral bacteria that affects the neurons. That creates a lot of swelling. It may not necessarily affect the muscle. All of this is really related to tetanus, which is really the muscle neurons. Meningitis is it's basically a viral infection of the entire central nervous system.
Wouldn't that target involuntary muscles?
Which ones?
Meningitis?
Certainly. They probably, and I don't know the specifics of meningitis, but they can affect both. So they can certainly affect your breathing and wherever it affects. These specifically are motor neurons. Yes?
What about like multiple sclerosis?
Oh.
Or myelin sheaths?
Oh, I've got good slides on those. So hold that question for that one because we talk about multiple sclerosis. So now that we know how things move in and out of the axons and how things are transported from the cell body to the axon itself, let's start to take a look at the overall structures of these, of the neurons, not just the axons, in terms of the information flowing into the neuron. And here you can see the first type. You'll see multipolar, or bipolar, and unipolar. And all this is related to the position of the dendrites. So looking at multipolar, you'll see that we have the most common type in the brain and spinal cord. If you were to take a look at those neurons, you would note that most of them are multipolar. And you'll note that information is flowing in from dendrites that can be spread out. So we've got multiple dendrites that are sending information to that cell body. And looking at the brain and spinal cord, remember I mentioned in the brain and spinal cord we had a bunch of a certain type of neuron. The term that I used were interneurons. Okay? And interneurons, central nervous system, as you guys illustrated earlier, we know that the brain and spinal cord, which is a central nervous system, which is the integrative center, is taking in lots of information, right? So it's got lots of information that's coming in from sensory organs. So naturally, there's going to be a lot of information. There can be multiple axons that are terminating on the central nervous system dendrites. So all these neurons are receiving information. This might be, again, simplifying it. This might be auditory information. This might be pH information of the blood. It's all coming in so that these interneurons can integrate that information. Now, looking at the next one, you'll see bipolar neurons. And here, bipolar just demonstrates that the dendrites and axons are on exact opposite sides of each other, meaning dendrites send information in, it'll collect information and send it in, and then back out. And that's just going to convey information through the cell body. So here where you saw dendrites as projections from the cell body, here you can see the dendrites converge all together into one dendrite stem, into the cell body, and come right back down the axon. And typically you would find these in our specialized sensory organs, our retina in the eyes, inner ear in the ear, and olfactory. Inner ear for balance. Unipolar neurons, on the other hand, only have one process. And it's one process that you can see off to the right here, where information just flows through. It doesn't flow through the cell body, it flows from the area that are the dendrites and also the axon. So here you will be able to see, here are the dendrites, here's the axon hillock, and then this becomes the entire axon, and here's our synaptic terminals.
So it never goes to the cell body?
Actually never comes through the cell body. So the cell body here in this case is really just meant to support and actually doesn't participate in the electrical conductivity of the neuron itself. So this is unidirectional. So here, just to recap, multipolar means it's sending tons of information through different sides of the cell body. Here you see the dendrite coming in and axon going out. Here you see the dendrite and axon going through and where the cell body is attached off to the side. Now, if we were to zoom in and take a look at special cells, we do have two types of cells that are also special as well, and that I'm going to be talking about. Purkinje cell and pyramidal cell. And you might see the term Purkinje, and if you have taken physiology before, You might see the term Purkinje also as Purkinje fibers in the heart. They're not the same. They're not to be confused. But what they do have in common is the term Purkinje. Because Dr. Purkinje was a histologist that first classified the structures. He was able to look at the structures of the heart, and he was able to look at the structures of the neurons. And what he noticed about the Purkinje cells, these cells he was able to find in the cerebellum. And what's classic about our Purkinje cells is that they have a very heavy dendritic tree. Most of the dendrites on here you can see that are heavily branched, and that's because our cerebellum is receiving lots of information. Cerebellum, as you remember, is all tied into motion. So this is all involved in kinesthetic type of activities, movement, joint positioning, etc. So it's taking all that sensory information and being sent to the cerebellum that's going to be processing that information. So naturally, it has lots of information to be receiving from different areas of the body, different areas of the brain that it could be sending information to, and to undergo that processing of information.
So someone that's in a vegetable state doesn't have those
I'm sorry?
working? If you're in a vegetable state, you don't have those specific neurons working?
It may not be, it's hard to say where the injury is, but in most cases of injuries where paralysis takes place, like that you're talking about, typically would happen in areas that are the longest of the neuron. So if you were to see where the longest part of the neuron is, typically the axon. So like the severing of anything, the nerves that come in, the severing of the nerves can cut the axons that communicate from the central nervous system all the way to its intended target. And depending on where that break is, if that break is somewhere near the spinal cord, it can be related to some of the other involuntary functions, such as the bladder, such as the diaphragm, the heart itself. Now, this particular example right here is an image from a scanning electron microscope where they used a fairly new sensor. And that allows them to really zoom in to take a look at the dendrites of the Purkinje cells. And again, these are located in the cerebellum. You can see that on top of the dendrites, you can see all these little mini dendrites. And it's very evident here that this allows the cerebellum to receive information from multiple sources. So now with that being said, now that you know the structures of the neuron, let's take a look and tie that into the functional classification from the example that we talked about earlier with the incoming car. So here you see that we have the neurons, the sensory neurons. Sensory neurons that have one function, is to transmit energy that takes place in the external environment or the internal environment of our body. It's conveying that information, that sensory information, back over to the central nervous system. So in the case of feeling cold, right? Or let's just say excess acids that are being produced in the stomach are being picked up by nerves, by pain sensory neurons that are transmitting that information to the central nervous system. Those pain sensors aren't saying, oh wow, that's a lot of pain. That's probably stemming from not only the stomach, it's probably caused by this. No, all it's doing is it's conveying pain information over to the integrative center, where it's going to take that pain information and say, if I'm feeling pain in the stomach, I'm also feeling pain somewhere else. The taste in my mouth is somewhat sour, so maybe this could be something that's going on. And then it's going to process that information, and it's going to result in the efferent pathway, the motor pathway. This is the decision that's made by the inner neurons. It's going to, again, our inner neurons doing the thinking, doing the decision making, the perception of information, the perception of pain. Those pain receptors are just saying, here's pain, that's all it is. Your inner neurons, your central nervous system is perceiving it. You're perceiving pain all the time. You're perceiving information all the time. I'm sorry, you're receiving information from your sensory all the time. You're receiving information about the temperature in this room. You're sensing information about how much food is in your stomach. The perception, though, is your awareness of it. And then lastly, like I said, the motor is the efferent pathway where it's going to send it to muscles and glands, its targeted organs. So we can divide those. When we talked about earlier, we talked about the voluntary actions versus the involuntary actions. And to kind of help tie in the sensory, integrative, and motor functions, we can take a look at the structures that are involved. As we mentioned earlier, you can see this is the case of a guy writing with a pen. And this is one of those things that is a learned task. But in this learned task, the learning isn't really the focus. It's the sensory receptor that's receiving information about how hard this person is pressing on that pen, which is going to send that information. We have these pressure receptors that detect how much pressure is being placed at that one spot. That's going to convey that information all the way through the sensory neuron that's going to make its way over to the integrative center, shown here in purple. Here you would see the sensory neuron, again something that we'll talk about in our peripheral nervous system lecture. The sensory neuron, you'll note that the cell body is outside away from the dendrites over to the axon. So this is an example of a unipolar neuron, where the clusters of the cell bodies are located in an area called the dorsal root ganglion, DRG. Again, we'll go over this in a later lecture. Now that is going to send that information over to the inner neurons. We're back over to either the inner neurons that are located in the spinal cord or located in the brain. And those inner neurons are going to interface possibly with other inner neurons. So there's a lot of information that's getting conveyed to different areas of the brain. The efferent pathway, otherwise known as the motor neuron, you'll see the motor neuron is going to be divided in from the upper motor neuron, that's going to take its place all the way from the brain, and again we'll talk about where, all the way down to that corresponding spinal cord that innervates the hands. So we're going to be able to target a lower motor neuron that's again sending that information from the integrative center all the way back out to the targeted muscles. So any feedback information that's sent in through the sensory, integrated into the central nervous system, and effectively performed by the motor neurons to the targeted muscles. In the involuntary side, this is something, again, we'll talk about later on. This is our classic knee-jerk reflex. So in this case here, you can see you see the quadriceps. You can see a patellar tendon hammer, the patellar hammer that's going to come down, and we're going to strike the patellar tendon. Have you guys ever seen this test being done before? This is a way of testing their reflexes, specifically with the spinal cord. So they strike the patellar tendon, which is going to cause the sensory receptors located on the muscle belly itself that's going to send afferent information through the dorsal root ganglion that's going to contain the cell body over to the gray matter of the spine. And the gray matter is going to contain the inner neurons. Again, something we'll talk about later. The inner neurons, the cell bodies are located in the gray matter. And that gray matter is going to send that information back out through the motor pathways to elicit a reaction. One reaction that's going to be involving the quadriceps itself, and the other one involving the antagonist muscle, the hamstring. And you'll see that each of these target one muscle, and this targets another. So this is an example of a reflex pathway. And again, there can be another branch from this right here that sends its information back up to the brain for the perception, the conscious perception of that information. So with that, let's take a break, and we're going to cover the next 15 slides. And shall we take a break?